Okay, good morning, everybody. And this morning I'm starting a series of classes on viruses and especially on COVID-19. I've moved these up a little bit. I was going to do them later towards the end of the semester, but I've moved them up, particularly because of the current surge in COVID-19 infections occurring throughout the nation. But also because we're approaching the Thanksgiving holidays, and some of the stuff that I'm going to tell you, I really would like you to take to heart and bear in mind over the Thanksgiving holiday. Okay, so let's have a talk first of all about viruses and the nature of viruses, exactly what they are, and something about how they operate. This here is a cell of some sort, but it looks like a white blood cell actually, which has been infected with virus. And you can see the surface of the cell here is covered with these little yellow dots. Each of these little yellow dots is a viral particle. It's a virus. And what is happening is that the virus has been replicated inside the cell and is now leaving the cell. It's budding out of the cell. And these viral particles will then be liberated. In all probability, the cell will die because all of its functions have been turned into viral functions. It's been turned into a literal viral factory, and the normal functions of the cell have been lost. So let's have a think. Just what we hear that word virus so often, and we need to have a clear idea about what exactly we're talking about. Well, viruses really are nothing more than extremely complex biomolecules. They are at their very simplest. The simplest viruses consist simply of a strand of nucleic acid of some sort that has all the information needed to produce more viral particles. That little strand of nucleic acid is surrounded by a protein capsule. It's actually called a capsid in viruses. So you have a little strand of nucleic acid surrounded by a protective protein coat. That constitutes the very simplest virus, and there are many viruses that are just that. That's all. There are some complications, as we'll see in a minute. Some are a little bit more elaborate than that. So viruses, as you saw from here, viruses are extremely small. They're hundreds, if not thousands of times smaller than a bacterial cell. And viruses infect every living thing has its viruses. So for example, bacteria have many, many viruses that infect them. Fungi, plants, animals, all have viruses that infect them. But a virus is itself an obligate parasite. Viruses have no metabolic function, and they have no ability to reproduce outside of a host cell. They're absolutely dependent on their host cell for all of their reproduction. So they take them outside of the cell. There is nothing there that would tell us that what we're looking at is a living thing. The virus has no metabolism. It doesn't do any of the things that a normal cell does. It doesn't have any ribosomes. It doesn't have a cytoplasm. It doesn't have a cell membrane. Although some of them may have a membrane around them, as you'll see in a minute, it's not a true cell membrane. Okay, so outside of the host, they are inert particles. They have no life, and they are not considered to be living things. So this is a very simplified view of how a virus operates. First of all, there are free viral particles circulating in some way. Viral particles can be transmitted through the air. They can be transmitted in water. They can be transmitted directly, blood to blood, body fluid to body fluid, etc. But the free viral particles need to find a host. And viruses are, in general, extremely specific about what host they can infect and what cells in that host can be infected. So the virus needs, first of all, to attach to its host cell and then to gain entry into the cytoplasm. It has got to access all of that cell machinery in order to reproduce. Once inside the cell, the virus uncoats and releases its genetic material. We'll hear that, I keep saying genetic material, hereditary material, because viruses can have various forms of nucleic acid. They're not necessarily DNA. It can also be RNA in various forms. Whatever it is, the hereditary material is released into the cytoplasm. Very often, it will become part of it'll integrate itself into the chromosomes, etc., of the host cell. Other times, it continues to operate inside the cytoplasm. It depends on the virus. Different viruses have different strategies. So once this hereditary material is released into the cell and begins to operate, what the virus does is it co-opts all of the functions of the cell, the normal functions of the cell. It stops most of the normal functions of the cell, except those that keep the cell alive. And it begins to force the cell to produce viral particles. So the virus now takes over the hereditary material of the cell, and the cell begins to produce two things. The first is it produces all of the proteins necessary to make up the viral particle. And the second thing that it does is it reproduces that nucleic acid, whatever it was, of the virus. Many copies of it. Produces all the viral proteins. And usually the viral proteins and everything assemble inside the cell, and then the viral particles are then released. There are different ways that they can be released. Sometimes the cell bursts to release the viral particles, but other times the viral particles actually bud out of the cell membrane. So they bud out without rupturing the cell. So, and that very often means the cell can continue to produce viral particles for a much longer time, continues to function as a viral factory until the cell is so damaged that it just ceases to function. Or else, don't forget that the immune system may see that the cell is infected and may kill it to try and stop the virus from being produced. So here's what we have so far. First of all, viruses are not cells. They are not alive. They don't have a cell membrane. They don't have metabolism. They don't have a cytoplasm or any of those things. They have a strand of nucleic acid as their hereditary material. But here's the thing, is depending on the virus, different viruses have different things. And they are incredibly diverse in their nucleic acids that they have. They can either have DNA or they can have RNA. And not only that, they can have bizarre types. So for example, there are two types of RNA that viruses can have. And they can also have single-stranded DNA or the normal double-stranded DNA. They can even have double-stranded RNA or single-stranded RNA. So they're very, very diverse in the structure of the hereditary material. But whatever it is, once the virus enters a host cell, it is turned into a form which the host cell machinery can recognize. So the host cell machinery can reproduce the hereditary material and produce all of the proteins and everything that we need to make up the structure of the virus itself. Okay, so let's have a look at the structure of some viruses and be prepared. They are incredibly diverse in in their structures. It depends on the type of virus that you're talking about. And for most of these, we as human beings have viruses that look like this. There's only one exception to that, as I'll point out quite clearly. So here is a very simple looking virus. This is one very similar to a virus that we are infected with sometimes, called an adenovirus. And here on the outside, this is the protein coat here, and it's this very neat geometric structure. We say it's icosahedral, and it's kind of hexagonal in cross section. These little round balls here are the protein units that make up this outer coat. And then on the inside here, there is DNA in case of adenovirus. But in other viruses could look like this, and they could have RNA. It depends on what virus we are talking about. So that's a really simple structure. But there are some things about it that we need to know. And that is that this outer coat here is really important, not only because it protects the hereditary material on the inside, but because this is how the virus recognizes the correct host cell to infect. Because these proteins actually interact with proteins on the cell membranes of the host or potential host cell. So these proteins, there are proteins embedded here in this coat that are able to interact with the host cell and bind the viral particle to the host cell. That's the very first stage of infection. And if that match doesn't occur, then the virus can't fix, and it can't settle, and it can't infect. So it has to find a host cell with the correct matching proteins that it can bind to. It's a very important stage in viral infection. And it's one that we're very, very interested in, by the way, in COVID-19, as I'll show you in a little while. So that's a really simple virus. We have viruses just like this that infect us and can cause illness. Here's another kind, another viral structure, and this is a helical virus, very often quite long and like strands. And in this case, you can have a look here and you'll see here the proteins are all laid down, in this case in a spiral, and the nucleic acid here is attached to these proteins, also in this kind of spiral. There is one very famous human virus that looks like this, and that is Ebola, E-B-O-L-A, Ebola virus, and here it is here, very long, thin strand like this, and Ebola characteristically, it has this twisted end like that. It looks like an old-fashioned carpet beater. So that's what they say. This is, it has this carpet beater shape, which is very typical. There are many viruses which form form filamentous structures like this. Here is another virus. Now this becomes important because this, although this is just a kind of a generic drawing here, many of the animal viruses take this form, and that is that in the inside here we have the nucleic acid, or whatever it is, RNA, DNA, whatever, protected by a capsid, a protein capsid. In this case, it's a filamentous protein capsid like this. But on the outside, surrounding the whole thing, there is an envelope. It's not referred to as a membrane. It's referred to as an envelope. And the envelope is derived from membranes from the host, whatever the host was for this virus. The membrane originated as host membrane, but its nature its function has been changed completely. You'll remember I told you that the function of cell membrane in a living cell is to allow conditions on the outside to be different to conditions on the inside. So we have outside of the cell, and then inside we have cytoplasm with its own conditions, and a boundary. The membrane is a boundary between the inside and the outside. Now the membrane, that's no longer performing that function. Instead, the membrane now has protective function. It protects this interior capsid and nucleic acid. It may also contain some proteins, enzymes, and things which are part of the virus, which quite often happens in animal viruses. But very, very importantly, embedded into this envelope here are many viral proteins. Here they all are here, shown quite simply here. These viral proteins, they can be very diverse. There can be many of them in this membrane. And their responsibility usually is, again, to bind the virus to the correct host cell. So it is these proteins on the outside that determine the infectivity of the virus. What can this virus infect? And I repeat, viruses are usually very specific. They have a specific host range, and that may be limited to one species. It may be limited to one group. For example, a human immunodeficiency virus, which looks something like this, only infects humans, cannot infect anybody else. And in humans, it only affects a very narrow range of cells. Those cells with a particular protein in their cell membrane can be infected by HIV, and that is it. Only human, only a restricted range of cells in the human. Other viruses, for example, just off the top of my head, rabies virus. Rabies virus is rabies is a terrible disease, a neurological disease, and the rabies virus can infect virtually any mammal. So it has a much broader host range than HIV. So it just depends on the virus as to what the hosts that it can infect, and in that host, what cells in that host can be infected. So this is just a generic kind of diagram for an enveloped virus with this helical filamentous capsid. This also, we'll also see that this capsid can be a geometric, icosahedral, or whatever as well. But I really want to show you this because these are coronaviruses, probably not COVID-19, not the virus that causes COVID-19. But you can see here, this is the membrane here, the envelope, the envelope derived from the host membrane, and embedded in it all these little bars that you see there. Those are all the proteins, the viral proteins that are embedded in this envelope. And this is why it got the name coronavirus, because it looks like a crown. If you look vaguely, you can see that it looks vaguely like a crown. That's where the corona part comes from. Okay, so here are the different forms again. So this is a helical virus forming filaments, and in fact, this is I'll tell you, this is a tobacco mosaic virus under the electron microscope. And this is important because the tobacco mosaic virus was the very first virus ever visualized. It was the first virus ever to be identified as well. This here is an adenovirus, and adenoviruses in human beings. The best known illness that an adenovirus causes is the common cold. It is one of 200 viruses that can cause the symptoms of the common cold. That is an adenovirus. Very simple. It has this protein capsid. Embedded in the capsid are spikes, or protein spikes, which allow this virus to find its host cell, correct host cell. Here it is here, also electron micrograph. This here is a typical enveloped virus. And in this case, this is influenza. It's the influenza. Again, you can see this. You can see the viral proteins all embedded in that membrane there. Influenza virus is one of the most important of all viruses that infect human beings. And there is a huge mortality associated with the influenza virus. If we look at our whole human history, it's been one of the most important diseases that ever affects human beings. It has a very, here is the envelope around the outside, but it has a very strange, influenza has a very strange genome. Its genetic makeup is a bit strange. It consists of RNA, but the RNA is broken up into a whole lot of different segments inside. It's in different units, which becomes important in a little while. And this last one here, this is one of my favorites. Now, this is a virus that does not infect animals. This is a virus that infects bacteria. And it is most extraordinary structure. It is actually like a little injection syringe. So here's the capsid here, up the head here. It's packed with the nucleic acid, that's DNA. And then all of this machinery here, and literally is machinery, all of this down here is machinery to find the correct host cell, bind to it, and then inject this DNA into the cell. This is a little injection syringe. Functions like a little injection syringe. And I'll show you a little bit more about it in a while. So this is a virus that infects bacteria, and we call that a bacteriophage or a phage. And bacteria have thousands. There are thousands upon thousands of identified viruses that that will infect bacteria. Okay, I think I've done. Just let me check. Okay, so I'm going to use that bacteriophage just as an example of how the virus gains entry into a cell, and then how it takes over the machinery of the cell. So this is we don't have this kind of virus. There's no virus exactly like this that infects us, but it's a good example of how viruses function. So first of all, let me go back to the structure of this virus. I told you there's the capsid with the hereditary material inside. In this case, it's DNA. This below here, this long filamentous part here is helical proteins. And in fact, the helical proteins, when the virus is free, those helical proteins are extended. They're like a spring that has been cocked. So it's extended like that. Running through the middle here, there is a tube. These down here, these they call tail filaments, but they look like legs. These are what determine, these are proteins which determine when the virus has found its correct host. And once those proteins there on the filaments there bind to the host cell wall, then this spring, then the legs collapse, and press the bottom of the tube against the cell wall there. And this spring then collapses. And it forces the tube right through the cell membrane, through the cell, into the cell, through the cell wall, through the cell membrane, and into the cytoplasm of the cell, and then spits its DNA into the cytoplasm. All of this elaborate machinery stays outside. It's just an injection syringe. That's all it is. And it spits the DNA in. Once the DNA enters the cell, it takes over the functions of the cell. in the case of these bacteriophages, what they usually do is they chop up the bacterial DNA into pieces, keep bits that are useful to them, and the bacterial cell now begins to produce all of the viral proteins. It produces all the viral proteins, and it reproduces the DNA over and over and over again, so there are lots of copies of the viral DNA as well. Those proteins, the viral proteins, actually assemble themselves inside the cell, and they assemble into the viral particles, and they package the DNA. And once they've done that, they burst the cell, and they are released from the cell, go on to infect other bacteria. So what distinguishes these bacteriophages is this elaborate structure. But in actual fact, this kind of cycle is very similar to the life cycle of most viruses. And there may be a few complications here and there, but basically this is the viral life cycle. Infect the host, take over the hereditary machinery, force the host to produce viral particles, package the viral genome into the viral particles, and then find a way to get out of the cell. Again, to repeat, the viruses they are all obligate parasites. They can only reproduce inside a host cell. They normally have a narrow host range, some broader than others. But one thing that needs to be said about human virus is many of our viruses that cause illness in us are derived from animal viruses and derived from animal populations. Now, there's two things that can happen. You can have direct communication with the animal infectious viruses from animals, for example. An example of that would be something like Ebola. Ebola is believed is resident in bats in Africa. And periodically, humans become infected from bats. And once humans are infected, they can infect other humans. So it is directly acquired from an animal population and then spread in the human population. Other viruses entered the human population a long time ago and have become specific to human beings, but they are derived from animal viruses. And a good example of that would be the human immunodeficiency virus. Okay. HIV is a virus which we can clearly see was derived from viruses that infect other primates. But it was a long time ago that entered the human population. And since then, HIV has become specific to humans. We can't even we can't reproduce the illness in any other animal. Lots of our viruses are in fact derived from animals. And when that is the case, that is called a zoonosis, plural zoonoses. But for example, probably one of the most important viruses that infect human beings, influenza, is a zoonosis. It is a zoonosis because we actually acquire it from animal populations. We acquire it from poultry and from pigs. And poultry, pigs, and humans are the specific hosts for influenza, and it cycles between those populations. COVID-19 is a zoonosis. It is, we believe, also acquired from bats originally. It may have passed through another animal, a host, to reach the human population, but it can be, very similar viruses can be found in bat populations even now. HIV, I've told you. Ebola, I've told you. Many of our viruses are zoonoses. Basically, this is just a summary for you. I showed you how the bacterial viruses infect the cell, take over the cell machinery. Well, our viruses, animal viruses, do exactly the same thing. This looks like a very complicated diagram. It's not. All that is happening is we have a free virus, an animal virus of some sort. It needs to fix to the host cell initially, and then it needs to gain entry. It needs to put its genome, its hereditary material, into the cell. What happened, don't worry about all of the machinations here. Just remember that once that genetic material has entered the cell, it needs to become part of the machinery of the cell. So there are various strategies depending on the virus. Some of them, their hereditary material stays in the cytoplasm. That is the case for COVID. In others, it becomes part of the host nucleus. It doesn't, it depends on the virus as to what strategies it adopts. But whatever it does, it takes over the machinery of the cell, and it gets in now, instead of the cell producing its normal proteins, for example, its normal enzymes, etc., the cell machinery begins to read the genetic material of the virus, and it begins to produce all the viral proteins, for example, and it begins to reproduce the viral genome, whatever it is. In this example, they've given you it's DNA, but it could also be RNA, depending on the virus. This viral DNA and the viral proteins are all assembled into new viral particles, and those then leave the cell by one means or another. Sometimes they burst the cell. That is called lysis. But in animal viruses, very, very often, they don't burst the cell. They bud out of the cell. And so the cell continues to function for a long time while it is producing viral particles. So that envelope around the outside of most animal viruses, that is derived from the membrane in the host somewhere. But remember that eukaryotes have many kinds of membrane inside their cytoplasm. Cast your mind back and you'll remember that we saw that the eukaryote cell has different organelles, each organelle with its own specific kind of membrane around it. We had endoplasmic reticulum, Golgi, mitochondria, all of these things had their own membranes. In the case of animal viruses, depending on the life cycle of the virus, the envelope around them can be derived from anywhere. It can be derived from the nuclear membrane. It can be derived from endoplasmic reticulum and from Golgi. It can also be derived from the cell membrane around the outside. It just depends on how the viral particle is assembled, and that would depend on the species of virus that we're talking about. In the case of COVID-19, the membrane, it is an enveloped virus, and the envelope is derived from endoplasmic reticulum and from the Golgi. So here is a typical animal enveloped virus. This is one that you have probably heard of, and this is the herpes virus. This is herpes simplex. This is the virus which causes cold sores around your mouth. And it consists, here's its envelope here. And embedded in the envelope, all of these here are viral proteins. So when the virus takes over the function of the cell, it causes the cell to manufacture envelope, and it manufactures envelope with these viral proteins embedded in it. And again, it's these proteins here that are going to determine what cells this viral particle can stick to and what it can infect. On the inside of the herpes virus, you can see here, this is in the herpes virus, there is this geometric icosahedral capsid like this, and they have a DNA strand as their hereditary material. They do, herpes virus also has some other proteins included on the inside of the envelope, but this is not cytoplasm. Okay, this is not cytoplasm, and it has no metabolic function once at all. Here it is again. And here's is the herpes virus. Herpes virus is a very interesting virus because most of the herpes viruses have one thing in common, and that is that when they infect a cell, here they are here it is here infecting the cell. You can see how the envelope sticks to the cell membrane there, and then it forces the cell membrane to open up so that it can spit in. There is the capsid. That's the capsid there. Here it's spat in the capsid there. And this, in the case of herpes virus, that makes its way to the nucleus. And it binds to the nuclear membrane and it injects the DNA into the nucleus. And in the case of the herpes viruses, that DNA strand, the viral DNA strand, becomes part of the genome of the host cell. And it can sit there like that for the lifetime of the cell and never do anything. But periodically, it will, these cells can be activated, and they then begin to produce viral particles. So this is characteristic for the herpes viruses, that they can be latent. They infect a host, and they affect many of the cells of the host, and they then can become quiet and they can sit for years and years silent inside the nucleus of the host, and then erupt and cause illness of some sort. This is the typical life cycle, for example, for cold sores, but for many other herpes viruses as well. So you know, if you've ever had cold sores, you get the cold sore, you suffer from it for 10 days or whatever, and it goes away, and then you don't have it for years, and then all of a sudden, the day before your first date with somebody, the cold sore suddenly reappears. And this is what has happened. The virus has integrated into nerve cells. That is where the herpes simplex virus goes to. And it just sits there quiet. And then every now and again, it erupts and it causes damage to cells as it reproduces. So here's what's really important about the herpes virus that I want to point out. When it becomes active, actually a lot of it forces the cell to make all of the viral proteins. The viral proteins are actually re-imported into the nucleus and are assembled inside the nucleus. Here's a new capsid with the DNA packaged here, and it then buds out of the nucleus. There it goes there, it's budding out, and it's acquiring the envelope that surrounds it. So what it's forcing the cell to do is to manufacture membrane with viral particles, with viral proteins rather, embedded in it. So it's taking over all of the machinery here of the endoplasmic reticulum. It's subverting the endoplasmic reticulum. It's making the endoplasmic reticulum make viral proteins, and it's making forcing the endoplasmic reticulum to make nuclear membrane with viral proteins embedded in it. There it goes, it buds out, acquires its membrane, and it then takes over the cellular machinery, and the viral particle is moved through from endoplasmic reticulum to the Golgi. Go back and revise what we did about the function of these membrane systems. The viral particles move all the way through the endomembrane systems, and they are then released. So they're actually released from the cells. There are two reasons why this, for instance, let's just think about the herpes simplex virus of cold sore. There's two reasons why you get a sore. The first reason is that the virus actually ends up damaging cells of around your mouth, and so this whole process damages them so much that they actually die. The second reason that you develop a sore is that your own immune system recognizes that this cell has been infected. Once it starts to produce these kind of viral proteins in membranes and everything, your immune system picks up that it's been, and your immune system begins to attack these cells that are infected. Go back to what we did about immunology and learn about cytotoxic T cells, because these cells get attacked by the cells of the immune system and get killed. And that's another reason why you develop that inflammation and pain and everything when you have an outbreak of a cold sore. I will tell you there are many other herpes viruses and allied viruses. So, for example, the virus that causes chickenpox does much the same thing. It integrates into your genome, becomes part of your nucleus, etc., and remains quiet. You have chickenpox as a child, you get over it, it remains quiet for your whole life until you reach the age of 40, 50, somewhere around about there, and then it can reappear, and it can produce a disease again. This time it's not called chickenpox. The second time it produces a disease called shingles. So these can go in, be latent for a very long time, and then reappear. This is just a picture of a capsid, like the herpes capsid, budding out. It's forced, in the case of herpes virus, it forces the cell to make nuclear membrane here with viral proteins embedded in it, and then it buds out of that, so that it acquires the membrane here with all these viral proteins. And here's an electron micrograph of exactly that process happening here. So this question of so many of our viruses being acquired from animals is an ongoing problem, and is in fact, we believe it is getting worse as the human population expands, as it is doing, and encroaches on natural areas, and we begin to encounter animal populations that we haven't had close contact with before. And we are recognizing that new diseases are continually entering the human population. Ebola is a very typical example. We know that from about the 1970s, we were getting reports of this terrible hemorrhagic disease that people were suffering from in Africa. But it is such a severe disease that it tends to be limited. People die very quickly. It is incredibly infectious, and small groups of people die very quickly, and then the disease disappears from the human population. But we are seeing, like with Ebola and with other diseases, that the epidemics, these epidemic outbreaks, are worsened, especially when people travel. So, for example, if people become ill, they'll travel to a city to go and get treated in a hospital or whatever, and actually end up spreading the disease. So these we refer to as emerging diseases. Ebola would be an emerging viral disease. And we are unfortunately probably watching it move into the human population, where it can be propagated continuously. At the moment, it enters the human population from animal populations and is then spread. But we are unfortunately probably going to see it become part of the human population. So it remains in a human we call it a reservoir of virus. At the moment, for Ebola, the reservoir is an animal reservoir. It occasionally enters the human population, but eventually it'll probably move into a human reservoir and be kept in the human population entirely. We'll no longer need to have seeding from animal populations for these epidemics to occur. There are numerous other viruses which we recognize as being emerging illnesses. And these you probably heard of as chikungunya virus and Zika virus. These are viruses which are of concern to us here in the United States. They're from South America, but we have the animals present in the United States which form a reservoir, and those are mosquitoes. So these are viruses which are spread by mosquitoes. Just to show you some. This is Ebola. Remember, I told you it has it's a helical virus, forms a filament, and here at the end you can see they usually have this twisted end. This is chikungunya, which is just an icosahedral virus. You can see, and this is the Zika virus, which is an enveloped virus. So the one group of viruses that we really need to talk about are the influenza viruses. Influenza is, as you know, is largely a seasonal illness. It can occur at any time of the year, but it peaks in the fall and the winter months. And it does so, we believe, because it is largely spread by droplets. It's spread by aerosol. People with flu, especially people who are just getting ill, they're busy developing symptoms, are infectious for the time before they become so ill that they go to bed. So they circulate around, they sneeze, they cough, and in fact, just talking, especially loud talking, will produce a fog of moisture droplets. We do it continuously. We don't notice it unless the lighting is particular and you can then see it. But whenever we are talking, we're producing a fog of these small droplets. Remember how small the virus is. A very, very small droplet produced by us would be of the order of nanometers wide, a fraction of the width of a bacterium in the case of the smallest droplets we produce. But that very small droplet can contain thousands, if not millions, of viral particles if that person who's producing it is ill. We believe that in winter, these droplets last longer and the virus remains infectious for longer because of the lower temperature and also because of the lower humidity in the air. These droplets circulate in the air and are breathed in. Very often, in fact, we will pick up droplets like on our faces, whatever, and we infect ourselves if we ever touch mucous membranes in the nose or the eyes. This is a very common route for infection with influenza or any respiratory virus. Now, the influenza viruses are very diverse, and there are influenzas which infect many different groups of vertebrates. But we recognize that there are three kind of families of flu, and there is one in particular, which is the Influenza A viruses, which are responsible for the epidemics which sweep through human populations. There is another family, Influenza B, which we also get every year, but it is less severe and it doesn't cause these kind of pandemics. The Influenza A viruses are the ones which are most dangerous to us. And the reason is that they infect pigs, they infect poultry, and they infect humans. And there are specific viruses that infect humans, specific viruses which infect pigs, specific viruses which infect poultry. But they can move between the three populations. It is possible to cross-infect between those populations. And every now and again, as a result of that, the genomes of the three get shuffled and produce a viral virus with an entirely new genome. It usually enters the human population from poultry, if it enters the human population at all. It enters from poultry. But it can be from a reshuffling of any of the viruses that infect poultry, humans, and pigs. When a new virus like that enters the human population, nobody in the human population has any resistance to it. And it can sweep through the human population, first as an epidemic, local to where it's originated, and then as a pandemic over the whole world. Now, the really important part of the influenza particle, what is that is the envelope around it, which determines what it can infect and who it can infect. And there are two proteins in particular which are embedded in that membrane, and these are hemagglutinin spikes and neuraminidase spikes. These are proteins. So this is H and N. And we know that there are several different kinds of H, several different kinds of N, and they can be shifted. Their structure, of course, is determined by the genetics of the virus. So the H and N, these can be shuffled around. And this is where you will hear this appellation, like H1N1 or H5N1, etc. It tells you which hemagglutinin protein they have and which neuraminidase protein they have. So I'll just tell you that what is actually happening is on the inside of the virus, there are 9, the genome is broken up into 9 different segments. And those segments can get shuffled if, for instance, let's say you have a pig that gets infected with a human virus, and the virus starts reproducing. And then the pig gets infected maybe with an avian virus, and the avian virus starts reproducing inside. Now the pig has 2 kinds of virus that it's harboring. And these 9 genetic elements from the bird, 9 genetic elements from humans can be shuffled around. It can start producing viruses with partial avian, partial human. And if that pig now produces a virus which can reinfect a human being, we could be in real trouble, because that could be an entirely new species of influenza virus made by recombining these genetic elements from bird, human, pig. If that now enters the human population and it can be passed from human to human, we could be looking at a new pandemic. But under normal circumstances anyway, influenza virus is dangerous, because it undergoes from year to year just the ordinary virus without any major reshuffling or anything. It undergoes continual mutation. It mutates very easily. And what happens is that it changes these slightly, the H and N, but especially the H. From year to year, it changes just slightly due to mutation. And that is enough that our immune system stop being able to see it. So this year you get a vaccination against a particular influenza strain. During the year, as it moves through the human population, it mutates slightly. and it mutates so that your immune system next year sees it again, it doesn't recognize it anymore. It's changed the protein slightly. It's not this kind of major change. It's small changes, but enough to hide it from the immune system. This is why we have to get revaccinated every year for influenza. But if the influenza virus undergoes one of these major reshufflings between pig, bird, and human, then it becomes completely unrecognizable. And we will have absolutely we have to start developing a vaccine to this virus, much as we have had to do to produce a vaccine to COVID. So influenza is an incredibly dangerous disease. It is a very, very serious disease. And yearly, it has a very high mortality, human mortality, just under normal circumstances from year to year. But when one of these entirely new viruses, influenza viruses, enter the human population, then the mortality can be enormous, absolutely enormous. So this, in summary, first of all, in year to year, there is a slight change in influenza, necessitating a new vaccine every year. But it's always just a modification of the previous vaccines. So this we call antigenic drift, the small scale changes. But antigenic shift is when we get a major change, either a completely new recombination, or quite often we acquire new viruses directly from poultry. So our association with poultry and pigs is the source of entirely new viruses. Every 20, 30, whatever years, it seems a new one arises, sometimes extremely dangerous, other times not as dangerous as at other times. The last really great epidemic, influenza epidemic, was in 1918. We have had flu pandemics regularly since 1918, but 1918 was called the Spanish flu. The misnomer had nothing to do with Spain. It actually probably arose in the United States and was spread as a pandemic because this was during the First World War, and soldiers exported it to Europe and then re-imported it from Europe to the United States. It was absolutely a devastating pandemic, and the number of deaths worldwide was about 50 million, and that is probably a gross underestimate. Close to 1 million people died in the United States, and remember the population of the United States was a fraction of what it is now. But what is particularly interesting, that I want to point out to you, here is a graph for 1918 and 1919, and this gives you a mortality rate per 1,000 persons. And just have a look and you'll see that it entered the American population and very quickly produced an epidemic with considerable mortality. But it then died out over the summer, and people became very lax. Towards the end of this year, people were doing much as we are doing now. They were wearing masks. They were starting to be very careful about hand washing and social distancing. People stopped like socializing and this sort of thing. And the influenza virus, as influenza viruses do, died out during the summer. But in the winter, an absolutely devastating epidemic hit the United States. And the mortality rates were just enormous. It died out by the end of spring. There was another outbreak, however, the next year in 1919. But interestingly enough, after that, it kind of bounced around like this, and then eventually just seemed to die out. It's probably a combination of people developing immunity to it, but also it appears that it may have actually undergone some mutations to become less virulent. So it didn't cause them high mortalities, but maybe was present in the population in a less lethal form later on. This here, look at this here, and it is chillingly reminiscent of what we are watching at the moment with COVID, except that for COVID, we had an increase in the summer there, but we are now somewhere here on this curve for the winter. There's one other thing I want to tell you before I finish for today about the influenza virus. Influenza itself is a serious illness, and just on its own, it can cause tremendous morbidity. That is, it causes tremendous amount of illness. People, if you have influenza, usually you become bedridden at least for a couple of days. It is not a common cold. It's not a bad cold. It has its own characteristics. But here's something very interesting about this influenza epidemic in 1918. And that was, if you look at normal influenza, the highest mortality is in very young, like infants and children, but also in the elderly. Once you pass the age of 60, if you get influenza, you are at risk of severe outcome. But this influenza epidemic, its highest mortality was in young people. It was young people, between the age of 25 and 40 was the highest mortality. And people in that age group died in a particular fashion. And that was that their lungs, it was observed that all very suddenly, the lungs would begin filling with fluid. They would develop a very, very high fever, and very often would die in a matter of hours. And it was complete mystery. Why did young people die in this way? Well, this has implications for our current epidemic, because we believe now that what was happening was that in some people, the immune response to the presence of the influenza virus was so powerful that it actually ended up killing the person. All of those responses, the lungs filling with fluid, the lungs becoming inflamed, the very high temperature, all of these things are signs that the immune system is responding too strongly to the presence of the virus, and it's causing illness like that. This is much the same as we can observe in some people who suffer from COVID-19. Okay, and I'll stop there. I'll complete this on Wednesday, and we'll discuss the coronavirus itself on Wednesday.